Hi everyone, thanks so much for tuning in. We're going to go back into the next chapter of notes the professor gave in microbiology. This is going to be epidemiology. And since this is going to be one of the chapters on the final for my class, I'm going to just skip towards the slides that actually have the professor's mark on them. And those are the ones that he's actually going to be pulling the questions from. So we're just going to probably go through them, the extra ones very quickly, but then our main focus will just be on those ones that are marked. Unlike last time where I just go and explain everything, this one we're just going to really hone in on those specific slides that the questions are going to be pulled from. So medicine before science, we have new ideas as well as old ones, new in quotation marks, from medieval Europe, and people used to think that the source of knowledge for these harmful events came from the Greeks as well as other religions like Christianity, where they blamed miasma or just bad air. And then they also blamed heavenly punishment. This all changes due to an event in Lisbon, Portugal on November 1st, 1755. And that's going to be an earthquake. This causes people to focus on the rational. Where we go from there, we get into medicine in the 1800s, where you have the four humors. You have melancholic, you have phlegmatic, you have the choleric, and then you have the sanguine, or the blood. Medicine then focused on balancing the humors. And then they also would have used bleeding, cupping, emetics, and enemas, as well as surgery. However, there wasn't any anesthetics, nor was it aseptic, so very crude modes of doing that. Alright, so now we're going to get into the actual study of epidemiology. First guest, who will be John Snow, a doctor in England in 1854 during a cholera outbreak. Alright, so he was skeptical of miasma, and he believed that cholera instead was caused by water. So therefore, he used the map to show that cases centered around the Broad Street pump. The Broad Street pump was where the town or these areas were getting their water, and all of a sudden, all around that water source, he had all these cases of cholera pop up. So this is an instance where they're using statistics to figure out the cause of the disease. So, this Broad Street pump, what was happening, a septic tank was leaking and contaminating the water supply. You could also look at Florence Nightingale. And she's going to visit hospitals and make assessments about the conditions and the nursing methods. During the Crimean War, which took place 1854 to 1857, she's going to go and take 38 nurses to Turkey to manage the Scutari Barrack Hospital. She's going to clean it, and she's going to add clean dressings to wounds. This was brand new at the time, and while she was there, the death rate did not decline. The Sanitary Commission then flushed the sewers and improved the ventilation. Sorry about that. Those are some of her impacts that she's given. In 1858, she was going to use statistics to show food and poor sanitation led to the deaths of soldiers from typhus. This is going to reform the army in Britain. And then in 1859, she's the first female member of the Royal Statistical Society and an honorary member of the American Statistical Society as well. Her legacy would be seen in hospital design, wherein nowadays hospitals will have systems of fresh air, clean drains, and separate wings. Management at the hospital would now involve, thanks to her work and her assessments, the management of the hospital would be based off of two principles. Number one, you can't keep patients longer than necessary. This is going to decrease the amount of those healthcare associated infections. And number two, the first requirement in a hospital is that it should do the sick no harm. Once again, the hospital should be a clean place. It should be very well sanitized. So these two principles are still used today as part of her legacy. We also have the Hungarian-born physician Ignaz Semmelweis. In 1818 and 1865, but most importantly, 1847. In the Allgemeine Krankenhaus in Vienna, let me write that out. Allgemeine Krankenhaus. That's in Vienna. He was the assistant in obstetrics, responsible for maternity service. And this is again related to maternity services. And his legacy is that when he was observing the mortality depended on who delivered the baby. This is one of his leading studies where he saw a 13 to 18% death rate or mortality rate when the baby is delivered by students or physicians, compared to the 2% mortality rate when the delivery is made by midwives. Therefore, he is known as the father of infection control. He was noticing that these mothers are dying from puerperal fever. which is caused by strep pyogenes. So therefore, he's going to require people to wash hands. He's going to make it mandatory in chlorinated lime water. And this is going to drop mortality rate to 1.3%. So that's from 18 to 1.3. That's a huge decline in the mortality rate. Therefore, he's known as the savior of mothers. In his studies, he was able to gather data to convince the administration to change. Alright, so that's basically like a background on epidemiology. Now let's get into the actual terms of the study of epidemiology, beginning with, what is epidemiology? It's a good place to start, I think. This is going to be the study of distributions and causes of disease in populations. An epidemiologist will collect, compile data about sources of disease and the risk factors. They're going to design infection control strategies. As well as predict the spread. They're also going to be using different disciplines, such as ecology, microbio, sociology, and stats. We saw with stats that John Snow, as well as Semmelweis, they are able to use stats to make some advancements. And then also psychology. So a communicable, sorry about that, my brain is still a little woozy, I don't know why. Got a lot of sleep. Oh well, so a contagious disease. This is self-explanatory, so I'm not going to say the definition of what a contagious disease is, but I am going to give some examples. Measles, colds, COVID-19, and we are going to determine the transmission via the interactions between environment, pathogen, and host. Control of any of these factors will break that infection cycle. So if we improve sanitation, antimicrobials, and if we vaccinate and increase sanitation, this will break that cycle of infection. Non-communicable diseases or non-contagious diseases do not spread from host to host. One example of these would be Legionella pneumophila in the water systems of buildings. All right, let's talk about the rates of disease in a population next. So we have the attack rate, This is going to be the proportion of susceptible who become ill after exposure. Incidence is the number of new cases in a population at risk during a specific time, and prevalence. This is going to be the total. Keyword there is total. The total number of cases at a point in time in a population. This is going to be cases per 100,000 people. So incidence is new, however, prevalence is total. More rates of disease. Vocab terms include morbidity. I know it has the word morbid there, like Morbius. Morbidity is the incidence of disease in a defined population. So incidence, again, that is going to be the number of new cases. Contagious disease have a higher morbidity. Of infected individuals that may transmit to several others. Mortality is the overall death rate in the population, and that's going to be the fraction of the people who die. Lastly, we have, on this slide at least, we have case fatality rate. This is going to be the proportion in a population diagnosed with a specific disease that dies. Ebola has a very high case fatality rate. Let's highlight these vocab terms here. We have a few more. We have more rates of disease terms, so I'm going to switch to another color. Endemic. So our first term on this slide. An endemic disease is constantly present in the population. Such as the common cold. Sporadic means few cases every once in a while. Epidemic means unusually large number of cases. And this could be introduced or endemic. An outbreak is a group of cases at a specific time and population. We just highlight all of those. And then a pandemic is an outbreak that spreads to several continents. So, of course, COVID-19 is a pandemic, and interestingly enough, you could actually classify it as multiple things. So let's go up here to my actual notes taken in class and the lecture was happening. COVID-19 is also endemic as of making this recording. AIDS is also a pandemic as well, so is the 1919 flu. So you could actually add it here to endemic. Right, so the chain of infection goes like this. So you have the reservoir of the agent, then you go into the portal of exit, then you go to transmission, then the portal of entry, and then the host. And then, of course, it's a chain, so then that host might be another reservoir, and then the whole thing continues. Okay, next, we'll talk about the reservoir of infection. This is the natural habitat of the pathogen. So this could be in or on an animal, human, or in the environment. And this is important in disease control to identify. So like if it's coming from mice, prairie dogs, the US has some regulations to stop those rodents, Human reservoirs, however, include the symptomatic infections and the asymptomatic. So symptomatic, this is going to be obvious. You're going to see those symptoms. Asymptomatic, I think you could guess what this one is. It's harder to identify because you have a carrier. And they might not show any symptoms. But they can spread to others. Such examples would be Neisseria, gonorrhea, in women, and 33% of the population will carry the Staph aureus. And again, remember that Staph aureus, some of that is MRSA, where it's just so happened to be resistant, and this is going to be found in the nose. Non-human animal reservoirs. So, animal reservoirs now include the zoonoses. This means that it exists in animals and then it transmits to the human. So, any to human. For example, plague and rabies. This is going to be more severe in the human. And then environmental reservoirs. These are kind of impossible to eliminate. And one of the difficult ones that we have to fight would be Clostridium. All right, the portal of exit. Change color. This is going to be a surface on the body or an orifice. The intestinal tract. Of course, that's going to be the feces. Respiratory. We are going to exit as droplets of saliva and mucus. The skin is going to just be the skin cells shedding off. And then genital pathogens will be removed through semen and vaginal secretion. All right, disease transmission, our next step in this crazy chain. Vertical. When we talk about vertical transmission, this is when it's mom to fetus or to child from breastfeeding. Horizontal transmission would be from person to person via air, physical contact, ingestion, or a vector. Direct contact. This is from the hands. Of course, that's kind of self-explanatory, but the main one is fecal. Oral transmission. That's also self-explanatory, but hand washing will prevent this as best as possible. This is considered the single most important measure for preventing the spread of infectious disease. Some pathogens, however, cannot survive in the environment, so they require intimate contact. And of course, the intimate contact would probably involve the STDs. All right, so now indirect transmission via the air. This is going to be the respiratory diseases. Particles larger than 10 micrometers are going to be trapped by the mucus. And then smaller go to the lungs, and they carry pathogens. From talking, laughing, singing, sneezing, and coughing, you're going to make these nuclei. And these are going to be difficult to control. The measures available include negative pressure and then HEPA filters. However, again, there's a difficulty controlling all those nuclei. Vehicle-borne disease transmission involves fomites or inanimate objects. These, of course, are tabletops, doorknobs, glasses, any of those. You also have food and water. As a vehicle-borne means of transmission, these could be contaminated. Eggs are going to be contaminated by the salmonella from the intestine. Likewise, meat is going to be contaminated with E. coli from the intestine of the animal when it's being butchered. Cross-contamination. This is when the pathogen is transferred from food to food. Municipal water systems can distribute the pathogen in a wide range. So we had an outbreak of Cryptosporidium in Milwaukee, Wisconsin, due to those large numbers of spread. Okay, vector-borne. So vectors, we talked about vectors up here as one of those indirect means. These are going to be living organisms that carry the pathogen. These are going to usually be mosquitoes, flies, fleas, lice, ticks, and they could be internally or externally. We have two methods. We have mechanical, which is when the fly lays eggs on some dung on the field, then the cows eat the cows will eat, get slaughtered and stuff. The fly will go onto the meat and then the person eats it. Or we also have biological, which is like a mosquito biting you. Let me look up actually the difference is. I had it up here somewhere. Let's check. Okay, so a fly will be using feces, so that's mechanical, and then biological, this is going to be when a mosquito is using like venom to transfer the disease. Okay, that makes a lot of sense. That's the difference. So venom, and then contaminating via being on feces and stuff. Okay, virulence. So this is going to be characteristics of the pathogen. We already went over virulence so many times. That's the ability to cause disease. However, there's a new blurb of information on this slide. Factors that allow pathogen to adhere, penetrate the host cell, thwart immune defenses, or damage hosts. So this is kind of saying the means the pathogen uses to attach or cause disease or evade the immune system. Infectious dose, we also went over that. That's the ID 50. That's the amount of pathogen needed to cause disease. Doses below that minimum may produce asymptomatic infection. The incubation period this is the extent of spread. For example, long incubation allows for extended spread. So, in this case, 10K individuals drank water with Salmonella typhi. And then the 10 to 14 day incubation allowed the spread of typhoid to at least 6 different countries. So that longer incubation period of the pathogen let it spread to wider range. This happened in 1963 in a ski resort in Switzerland. How do we get the immunity to the pathogen? Well, we have two modes here. We have previous exposure and immunization. Herd immunity is only when you're going to have more than 90% of the population sufficiently immune. And then again, that just closes out all of these potential hosts for that pathogen to infect. Drop my Apple Pencil. All right, so again, this needs to still place an importance in immunization. That's why vaccines are still so important. That's why antibiotic research is still very important, as well as hand washing, because you need to do all those things before you could get to the herd immunity. Antigenic variation. This is going to overcome some of that, unfortunately. Some examples would be, of course, bird flu and COVID-19, other sicknesses as well. General health would also be a characteristic of the host. So if you're malnutrition, if you're facing malnutrition, sorry about that. I do not know what's going on this morning. I'm still very sleepy, even though I had a good sleep. Malnutrition, overcrowding, and fatigue. These would also make a host more susceptible. Developing world is more susceptible, unfortunately, due to all the poor food and sanitation. That's why it's important that we make that a highlighted issue in today's discussions and policies to help make sure everyone has the sanitation and the tools that they need in order to protect their well-being and health. Sorry, I keep fumbling words. I don't know. I must be suffering from some fatigue still. I'm still, pretty tired. I'm still mumbling and fumbling words. I am so sorry. I apologize. Okay, age. So usually you hear, age is a factor in determining if the host will get a disease or not. So usually very young people and the very elderly are more susceptible. This is due to the immune system not being very developed in the young. It's going to be worn out. And then the elderly are less likely to update immunizations as well, fortunately. Gender does play a role in this. There are certain diseases that a certain gender could get and the other gender cannot, or another gender may get this, or both genders may get this, or all the genders will get this. It just depends on which frame you're looking at. We're all human beings, so and just keep looking out for each other, put yourself in the other person's shoes. And so women more likely to develop urinary tract infections. And this is due to the urethra being shorter. And so therefore microbes are more likely to ascend it. The pregnant women are unfortunately more susceptible to listeriosis as well. There's also a factor of religion and cultural practices. a lot of religions are going to, or cultures are also going to be consuming raw fish. That's going to, of course, increase exposure. Breastfeeding will provide protective antibodies to the infant. Genetic background. So some people have natural immunity, some people do not. It varies. Specific receptors may differ. For example, lack of receptor in red blood cell yields. Sorry, I am still fumbling words. Darn it! Oh my goodness, I am so tired, and yet I got a lot of sleep. For an example, a lack of receptor on the red blood cell yields an immunity to Plasmodium vivax. Alright, so the factors of the host are going to be herd immunity, general health, the gender. So if the person, again, we talked about the physicality, if, like the women having a shorter urethra, that exposes it to microbes to ascend. And that goes for all the genders. There's differences in the pathogens that, unfortunately, they're going to suffer from or be exposed to. Religious and cultural practices, and then the genetic background. Characteristics of the environment are going to be temperature, water, nutrient supply, radiation, and the availability of light and O2. Crowding and poor sanitation, another factor in the environment. Endospores will survive. And then there's also the exposure to antibiotics, which we've covered before, or the misuse of antibiotics, and then you're creating all these strains, and then they're resistant because, natural selection and artificial selection just cause all that mess. All right, now let's talk about some things related to statistics. So we have descriptive studies. These are going to be related to the data collected following outbreaks. And so I'm going to move this down. Whoa. We have more room. So we're going to look at the person. So again, all of those characteristics. Age, gender, ethnicity, occupation, personal habits. They may all yield clues about the risks. We're also going to look at the place. This is going to be the location to help pinpoint the source. And we're going to be looking at clues for the potential reservoirs, I should say, and vectors, etc. And then also the time. So a propagated epidemic would be a slow rise in cases. I'm going to highlight this one because that seems most important. Slow rise in cases suggests that the disease is spreading. This is going to have the first case called an index case. I'm going to put in 1 as the I, so we know it's the first case. Then we also have the common source. This is when the rapid rise in cases would equal to a single source of the pathogen. This is like John Snow with that cholera epidemic in England. You notice that there's a rapid rise of all these cholera cases around a single area. So then he's able to say the what was it? The pump was the cause of that. The Broad Street pump. There we go. Right on it. We also have these national disease surveillance networks, such as the CDC. And they're going to support the disease labs. They're going to collect data on notifiable diseases. I'm going to go back up here. I probably wrote down the definition for that somewhere. Where did I write it? Oh, I guess I did not. They're going to collect information on those, though. They're going to publish morbidity and mortality weekly report. And then again, morbidity, if we go back up here as a little bit of a review, morbidity is the incidence of disease or the new cases of disease in a defined population, and then the death rate, of course, is mortality. So, cases, and then the death rate. We also have public health departments in each state. They're going to mandate which diseases, are reported, Going to also have other components of the public health network. There's going to be schools reporting absences. The hospitals are going to report on isolating pathogens of importance. Such as, again, those escape pathogens that we went over last time. The news media is going to also alert the public to the presence of infectious disease. Or they're going to try to, and people don't listen to them, Oh well. Hopefully that more people actually listen to the experts. Worldwide disease surveillance. So we have the WHO, World Health Organization. We're going to have 4 main functions. We're going to provide guidance in health. Number 2, they're going to set global standards. Number 3, they're going to cooperate and strengthen national health programs. And number 4, we're going to develop and transfer appropriate health technology. Especially, I would hope, to developing countries and the areas that are facing the most severe environments that are causing all these diseases. Hopefully those are going to be our priority. We're also going to give education and assistance, as well as disseminate information. Such as the weekly epidemiological report, healthcare-associated infections will be our last topic. These are going to be acquired when getting treated in a healthcare setting, and the patient didn't go there for it. So, like, if you get salmonella at the hospital, you didn't go in the hospital for salmonella. You came in there to get, like, maybe another medication. You just so happened to get it, unfortunately. This is going to be one of the top 10 causes of death in the U.S. They're also known as hospital-acquired nosocomial. We're going to have this problem in densely populated hospitals with susceptible people. So all those patients, basically, is going to be a great risk, an unfortunate one too, where resistant virulent pathogens exist. So we also have these. Pathogens amongst all these susceptible people. About 10% of the patients will get one of these healthcare associated infections or nosocomial infections, and 2/3 of them are from the patient's own microbiota. And that is a scary fact. Alright, that's going to be everything from that general epidemiology chapter. Thanks so much for tuning in, and please do something nice for someone. Take care.